Briefly, a taxonomy of sources of uncertainty. I have a presentation, sneak preview. Yesterday, I asked Robbie Holdaway in New Zealand if I could show his results, which are in review right now, because I think it's relevant to what many of you are interested in, propagating uncertainty for the country of New Zealand. But most of my talk will be about the Monte Carlo propagation of uncertainty in forest biomass, which is now an effort of QUEST, Quantifying Uncertainty in Ecosystem Studies, which is a research coordination network I'd like to invite all of you to join. And if I have time, I'll address aspects of uncertainty and some other components of ecosystem studies. This classification of uncertainty is one that Mark Harmon developed. He's on the steering committee for QUEST. So I expected someone to put up a slide like this, but I haven't seen one yet. There are many ways to categorize uncertainty, but one is to think of natural variability. So this is where we have sampling error is due to real variation in the landscape or over time. We can't reduce that by improving our sampling, but we can better quantify it. Knowledge uncertainty, we've spoken about quite a bit about measurement error and about model error. And model error includes selecting the model as well as describing the uncertainty in the model parameters or the model predictions. Okay, I'm going to switch here. I couldn't paste in the slides because the backgrounds were different, so I need to switch to New Zealand. So Robbie Holdaway sent me these slides because this work has not yet been published, but I saw it 6 months ago maybe, and he recently submitted it for publication. I thought this was relevant to what some of you are doing in your countries. They have a grid for sampling their forest resource, and what they did that I thought you would be interested in is they sent out 3 teams to independently measure 7 of the plots. These are the 7 plots. They're located in the North Island, and these are their methods. So they chose 7 of their plots. They put the teams under time pressure like they are in real life. They used real compositions of the teams. They have 1 botanist on each team. They have to identify species. And then they compared the results for those 3 teams. And these are the 7 plots. So you can see what sometimes the 3 teams are in close agreement, and sometimes they're not. And that disagreement was not very predictable. So he said, the one they thought would be really hard to get right, which they call the horrible, wet, steep plot. They had very good agreement, and this was, the worst one was one that they thought would be easy. So the 3 teams differed by 1 to 36% in the carbon stocks estimated on those plots. This coarse woody debris is a problem, in part because one team will say that's coarse woody debris, and another team says no, that's still a tree. When does a spar become a stump? So here are some of the measurement sources that I know you're interested in. Their uncertainty in tree diameters was 5%. Uncertainty in heights was 21%. They use species-specific height equations and species-specific wood density. So identifying species is another source of error that turned out to be very small. Each team had to establish the plot. So there was a 5% error in the plot area, 6% error in number of stems. So is a stem in or out of the plot, or do you miss a stem or double count a stem? And so number of standing dead stems, there's fewer of them and they're harder to judge. So there's a higher uncertainty in standing dead and in the coarse woody debris. They propagated uncertainty in the allometric equations, and I'll talk about that more later. I will note that they used equation number 5, which is the prediction interval, banda de predicción. Although when I visited him in July, he was using is that right? No, I asked Steen yesterday to help me get these in English. They were using the prediction for individuals, which is, I convinced him to use the prediction of the mean. There's an issue we'll talk about more later, confidence interval. So then they scaled these, they used a Monte Carlo to scale all these sources of uncertainty up to the national level. And these are the results. And also for change over time, because they have a 7-year remeasurement for 245 of their plots. Oh, thank you. So the uncertainty in stocks was small. This is 1% uncertainty. Altogether, most of that is coming from sources of measurement error. Model error is not distinguishable from 0. The error bars represent the uncertainty in the uncertainty estimates. You all appreciate that. So most of these are not significantly different from 0. And then when they went to the uncertainty in change over time in carbon stocks, there's a much higher percentage uncertainty, and the model error goes away, which I'll talk about again later in my own example. I think I'll stop now and ask for questions about this study. I am not a statistician. Many people have stood here and made a true confession. I am not a statistician. I am an ecosystem ecologist. I began my PhD research 30 years ago at the Hubbard Brook Experimental Forest to do a phosphorus budget, back when ecosystem budgets had no error. So this is an example. This is not my budget. This is the first nitrogen budget that was published for Hubbard Brook. And it represents a huge amount of work, quantifying many pools and fluxes. There are no uncertainties in any of those pools and fluxes, but they compared all the inputs to all the outputs, and there was 14.2 kilograms per hectare per year unaccounted for, which they called nitrogen fixation. So it always troubled me that we didn't know 14.2 plus or minus what. So 14.2 was obtained by comparing all the sources to all the sinks. The sinks being biomass, precipitation is a source, stream water is a sink, the forest floor and the soil could be sinks or sources. I'll come back to some of those if we have time, but mostly I'll be talking to you about how we estimated uncertainty in the biomass. So 5 years ago, in 2008, the Forest Soils Group of the Soil Science Society of America proposed a symposium on nutrient budgets. And I said, someone needs to do uncertainty in the nutrient budgets. And they put me down for a presentation on uncertainty in nutrient budgets. So fortunately, I was on my way to Woods Hole to work with Ed Rastetter on ecosystem nutrient cycling. But I told him I wanted to propagate uncertainty in an ecosystem budget using Monte Carlo. And he said, you can do that in Excel. That was how I got started. When I started, I did not even know the rule for propagating uncertainty in a sum. You all know these things now. So I thought this could be relevant. We sum the nitrogen in biomass in all the components of all the trees in the ecosystem. Each of those pools is a product. So mass times concentration, and Ed Rastetter didn't know the product rule, but we found the product rule. But it gets worse because, as you know, the masses are estimated with these log-log regression models. The way we do it at Hubbard Brook, we estimate mass from parabolic volume, and parabolic volume is a function of height and diameter, which is another log-log regression. I found this statement in Whitaker's publication of his allometric equations saying that he didn't know how to propagate uncertainty. So it's not true that that wasn't known in 1974, and you can do it analytically with Gaussian error propagation, and there is an example on the Quest website of a Gaussian error approach to coarse dB conducted in Excel. We didn't want to do that. There are equations for each species, for each tissue type, so we used a Monte Carlo. A Monte Carlo uses random sampling of the inputs to any calculation. So in this case, this is a flowchart notation. We have a tree inventory. We have the allometric equations. Each loop through the Monte Carlo, we sample the tree diameters with measurement error, and we sample the equations with the uncertainty in the regression. And we go through calculating the height, the parabolic volume, the mass of each component, the nitrogen content, add them all up, and if we're simulating plots and sampling error, we do that for each plot and go around this loop until we're sufficiently confident of our uncertainties. I was happy yesterday to see these slides from Fernando describing the uncertainty in regression. So I sent him an email and asked if I could find them, and then Andy said, they're on the laptop. So I'll make sure I have his permission before I give you these slides. So this could be a regression model of tree height or biomass. And what he showed was these two equations that I was asking how to describe. And I learned that this one is called the banda de confianza. And this one is called the banda de predicción. Is that right? Anyway, so this is the equation we want to use if we're describing the mean prediction of the model, and this is if we're concerned in estimating an individual. And he had a graph. So if these were our individuals, this confidence interval here would be the uncertainty in the mean, and the uncertainty in predicting an individual is wider. So in the paper that I published following my 2008 initial effort at this problem, there's a section on selecting the error terms. And I put this up because this is one of the hard, implementing the Monte Carlo is not hard, but it's easy to make mistakes. And one of the mistakes is whether you need to use the uncertainty in the mean or the uncertainty in an individual. And when we're on a forest plot, we care about the uncertainty in the mean. And there have even been cases, and John Battles did this too, of using this equation, the standard deviation in the regression model in some of these Monte Carlo approaches. There are many things that claim to be some aspect of uncertainty in the regression, and you have to think about using the right one. So you can do this in Excel. How many people have done this already, a Monte Carlo? I don't recommend Excel. If you want to do it more than 100 times, you'll get tired of doing it in Excel. But Craig suggested I show this because you can see the steps better than when you push the run button in R or SAS or some more sophisticated programming language. So we have these tables of our parameters for the models. This example is the height parameters. So the log-log regression has 2 parameters and an error term. And we randomly generate an estimate of error based on this error term. That's the standard error of the regression, and then we need to look at the diameter of the particular tree that we're predicting to get the correct error term for that. So our Excel formula uses VLOOKUP to get each of these parameters and the error term. And it's important that the random selection of the parameters applies to the entire Monte Carlo iteration. If the errors were sampled independently for each tree, they would all cancel out. And we know that if you had an infinite number of trees, your model uncertainty should not be 0. So once we have our height and calculate our parabolic volume, we again use a lookup function to get the parameters, the model predictions for the biomass of the wood and the bark and the branches and the leaves. We need to do that because we're multiplying by nitrogen concentrations. Your calculations can be simpler if you're estimating carbon. So we have nitrogen concentrations with a standard error of the samples to estimate the nitrogen contents of the bark and the branches and the leaves and the stem and the roots. So in Excel, my technician called this an epic formula. So you're looking simultaneously, or in one formula call, you're looking at all those tables of random numbers. Craig told me that in R, he generates all the random numbers ahead of time, and it's faster to look at them that way. So in Excel, we are adding up 4,000 trees in the watershed and copying those sums, whether it's the biomass, the nitrogen in each of the components, or the nitrogen in the whole ecosystem, and pasting it into a results file. So this is why you're going to see results that have hundreds, not thousands, of iterations, because you paste. Every time you paste, Excel updates all your random number calls. So each iteration is one paste. So we analyze the results. We can ask for the mean of all our iterations, the variance. We could calculate a coefficient of variation. So here's my example of nitrogen in biomass for the Hubbard Brook ecosystem. This is just 100 iterations, we can describe the frequency distribution of those estimates, and the 95%, is this a point? The 95% of these iterations fell within 54 kilograms of nitrogen per hectare of the mean of 611. That was pool size. But from the point of view of the missing 14.2, we are interested not in the pool size, but in the change over time. The same is true for carbon accounting. So again, it's important that the model uncertainty be sampled simultaneously for both points in time, just like we have to do it for all the trees in one time. And the uncertainty between these two measurements can be less than in a single measurement. So here's 100 iterations of nitrogen in biomass from the 1997 inventory, and you can see the uncertainty is 50 kilograms per hectare. In 2002, there's a similar spread, plus or minus 50 kilograms per hectare, but in every case, the 2002 estimate is a little bit higher than the 1997 estimate. So if we look at the frequency distribution of the estimates of change over time, the uncertainty is only 5 kilograms per hectare over 5 years. So 1 kilogram per hectare per year is the uncertainty in the change. If I report it as an uncertainty, as a percentage uncertainty, which was the way the New Zealand results were illustrated, it would look like a large change. But in kilograms of nitrogen per hectare, it's a smaller uncertainty than the stock uncertainty. The point has been made in this workshop that comparing sources of uncertainty helps us identify where we should put effort to reduce uncertainty. Oh, this doesn't show very well. This is an illustration of a not very useful approach, which is a sensitivity analysis, which was what I included with my first phosphorus ecosystem budget. And it varies each of the inputs by 50%, which is not very meaningful if some of the inputs are more uncertain than others. We could also look at the uncertainty in the model predictions, but again, some of these are more important than others. So if we have a high uncertainty in bark, if we have higher uncertainty in the estimates of bark than wood, when you multiply that out by how much bark there is in your ecosystem compared to how much wood is in your ecosystem, the wood will be more important. So this is the amount of the pool size in my ecosystem, the coefficient of variation in my models, and then if I run it through my Monte Carlo, I can see that bark is not as important in stem wood, although the model uncertainty was higher, and that branches are the most uncertain component of my budget. So maybe we should be including more trees in our branch equations than we need for our stem equation. This is the same analysis applied to nitrogen content, which is of less interest to this audience. So I have a summary slide about sources of uncertainty in forest biomass. Our measurement, our DBH measurements, according to John Battle's crew, is a very small source of uncertainty. Sampling variation, if we apply this across the Hubbard Brook Valley, is a larger source of uncertainty. Depends how many plots you put out. Nitrogen concentration estimates contribute about 3%. Our allometric equations came to 5%, and that does not address the problem of model selection. I use the same number from the Nelson paper of 20 to 40%, and this is a local example of different predictions by different equations for sugar maple taken in New Hampshire. So to summarize advice on biomass uncertainty, be careful of the selection of error terms, whether you are predicting an individual or predicting a mean in a stand. If you're adding up trees in a stand, you're interested in the mean. And then applying the error terms appropriately. So simultaneously sampling error in your regressions for any iteration of the Monte Carlo across all trees or across all time periods. And finally, estimating uncertainties can help set priorities for monitoring and research. Someone told me yesterday the plan to first estimate carbon and later think about the uncertainties misses the opportunity to use those uncertainties to develop your monitoring plan. QUEST is an acronym developed by John Campbell, one of my QUEST leaders, quantifying uncertainty in ecosystem studies. Certainly we are interested in uncertainty not just in ecosystem budgets. I was happy to hear that I have a link here. Craig told me he noticed that this workshop was referenced on the QUEST website. So I'll show you the QUEST website. So there it is, QUEST News, Workshop, Error Propagation for Carbon Estimation. If you click on it, it links to the website. I think I was speaking with Jim about this Canadian effort to propagate uncertainty in carbon estimates, but they used the uncertainty in the individual in making their calculations. We have, if you have relevant papers and send them to us, we will add them to our bibliography. If you have sample code, we'll share it. Initially I put those Excel spreadsheets for the nitrogen budget on my website, and people were using it before QUEST was ever funded or this website was developed. So, how much time do I have?
2 minutes.
I'll just show you some summary slides for other aspects of the ecosystem that QUEST investigators have been working on. You can be glad you don't have some of these sources of uncertainty. Measurement, error in precipitation, spatial interpolation is a topic you're interested in. So John Campbell compared different methods of interpolating precipitation volume to characterize these watersheds given the points at which precipitation is measured. And that turned out to be a very small source of error, partly because the sampling density is so high. Similarly in streams, in contrast for streams, precipitation collectors integrate over time, but we have to interpolate spatially, whereas a stream integrates over space, but we only collect samples to describe stream chemistry weekly, so we're interested in temporal interpolation. So we can quantify model selection error, different ways of doing that interpolation, different sources of measurement error, and again, natural variability is a big source. A wet year is different from a dry year. Soils. I know you're interested in soils. We have high measurement uncertainty in rocky soils and high spatial variability. So if we have repeated measurements of forest floor, carbon and nitrogen, and we can put a regression through those points and describe the standard error of the regression. And the uncertainty in regression, remember I wanted to put an uncertainty on 14.2? It's 22. The uncertainty in the slope is 22 kilograms of nitrogen per hectare per year. And the mineral soil is worse. Measuring quantitative soil pits before and after a whole tree harvest, they couldn't detect a difference, but the uncertainty was 53. So there's the answer, and I can use the error propagation rule for a sum. 14.2 plus or minus 57 kilograms per hectare per year. That's because of the soils. So if you can frame your question to exclude soils, you have a more tractable problem. So we're writing a paper about the uncertainty in just comparing precipitation inputs to stream water outputs, and that uncertainty would be only 1.7. I'm out of time. Summary. Step through the calculations to identify your sources of uncertainty. Think about knowledge uncertainty and natural variation. It's not hard to do a Monte Carlo, but it's easy to make mistakes. And we have all experience with making many mistakes. And if it's not in Excel, it's easier to fix your mistakes and run it again. You can get help, provide advice, join Quest. And we have a Gmail account, quantifyinguncertainty@gmail.com, where you can send us an email, tell us you want to be on your mailing list, or send us questions or suggestions. Thank you.
Secretaría de Medio Ambiente y Recursos Naturales.